dog's paws just moved. A small twitch, then a second one, more deliberate this time. Their eyes drift beneath closed lids. A low sound escapes. Not quite a bark, not quite a whimper. Something caught between the two. You've seen this dozens of times. You've probably assumed it was nothing specific, a reflex, a muscle memory firing at random, the biological noise of a resting body. But when neuroscientists attached EEG electrodes to sleeping dogs and looked at what the brain was actually doing during those moments, they found something nobody expected. The brain wasn't resting, it was running. The assumption is reasonable. Dogs sleep roughly 50% of every 24 hours, and we tend to read that stillness as absence. They're offline, recharging the way a phone charges on a nightstand, doing nothing useful while it fills back up. When pressed, most people would say their dog probably dreams in some basic sense. Maybe fragments of smell, a familiar warmth. Sensory static without real content. A simplified, dimmed version of what the human brain does. But here's the problem. The sleeping dog brain doesn't simplify. It doesn't reduce. In the specific phase of sleep where dreaming occurs, the canine brain displays electrical activity that is almost indistinguishable from a fully awake, actively alert animal. Processing, firing, reconstructing. Not switched off, not dimmed. Running something with considerable precision. When researchers analyzed EEG recordings of sleeping dogs, they identified something called sleep spindles, rapid bursts of synchronized brainwave activity in the 9 to 16 Hz range, appearing during non-REM sleep. These aren't background noise. Sleep spindles are an active neurological mechanism, a self-generated shield that blocks incoming sensory data from reaching the cortex, specifically so the hippocampus can consolidate new information into long-term memory without interference. A dog sleeping through a thunderstorm isn't ignoring it. Their brain has actively routed the sound away. A study examining EEG transients in sleeping dogs found that animals with higher spindle activity during non-REM sleep showed measurably stronger performance on learned tasks the following day. The spindles predicted the learning. The sleep was doing the work. But the more interesting finding is what happens approximately 20 minutes after sleep onset. The spindles thin out. The brainwave signature shifts entirely. What appears on the EEG is low-voltage, mixed-frequency activity, the electrical fingerprint of a fully engaged, visually processing brain. Dogs spend only 10 to 12% of their total sleep in this REM phase, compared to 25% for humans, a narrower window. But the intensity packed inside that window is, by every neurological measure, equivalent. In those compressed minutes, something is being projected onto the visual cortex with enough resolution that the body begins responding as if the events were real. But the more interesting finding wasn't the EEG signature itself. It was what generates the images. Inside the brainstem sits a small structure called the pons. During REM sleep, the pons produces a specific class of electrical spike, PGO waves, short for ponto-geniculo-occipital. These spikes originate in the pons, travel through the thalamus, and terminate in the primary visual cortex. They begin firing 30 to 60 seconds before REM sleep technically starts, before the dreaming is underway. They don't respond to external light. They don't require open eyes. They are a self-contained internal projection system, burning imagery directly onto the visual cortex from the brain's own electrical output, without any input from the outside world. Your dog sees things that aren't there, and the brain cannot distinguish that constructed image from one produced by actual light entering actual eyes. Synchronized with hippocampal memory replay, These PGO waves project reconstructed scenes with enough continuity that the motor cortex begins sending movement commands toward the limbs. The paw twitch is not a reflex. It is the beginning of a real movement, one the rest of the system then has to cancel. Compare that to what happens when the cancellation system fails. The pons doesn't only generate the dream, it manages the off switch. At the onset of REM sleep, specific cells in the pons release inhibitory neurotransmitters, primarily glycine and GABA, down the length of the spinal cord, paralyzing the major skeletal muscle groups. Only the extraocular muscles and the diaphragm are exempt. Under normal conditions, the system holds completely. In older veterinary research, scientists temporarily deactivated this inhibitory circuit in sleeping dogs. Without the chemical restraint, the dogs stood up and physically enacted their dreams. A pointer froze mid-room and pointed, rigidly, precisely, at nothing visible to anyone but the dog. A Springer Spaniel flushed birds that existed only in the visual cortex. A Doberman stood at the room's perimeter and guarded against an intruder nobody else could see. Each dog performing a behavior specific to their breed, driven by a scene their brain was treating as entirely real. The off switch was the only thing separating the dream from the room. This system exists because of what dogs actually are. Domestic dogs are the only animal domesticated before the invention of agriculture, before farming, before cities, before written language. Roughly 15,000 years ago, the wolves most tolerant of human proximity began a slow genetic divergence. In 2017, geneticists analyzing chromosome 6 in domestic dogs identified specific insertions in a region associated with hypersociability, a biological drive toward human-directed contact that does not exist in wolves. The motivation to be near us isn't learned behavior. It is written into the genome, selected for across thousands of generations, because closeness to humans improved survival outcomes for the animals that carried it. What this means for the dreaming brain is neurochemically specific. The hippocampus, the structure responsible for episodic memory and spatial mapping, doesn't consolidate all experiences with equal weight. It prioritizes by emotional intensity. And the neurochemical that marks intensity in the bonding context is oxytocin, a neuropeptide your dog's brain releases in precisely the same way your own brain does during close physical contact and proximity. When the hippocampus selects what to replay during REM sleep, it selects for the experiences most saturated with oxytocin signal, the highest weight encounters, the most bonding dense moments of the preceding hours. Your nervous system and your dog's run the same molecular reward architecture. Not approximately, the same pathway. And during REM, the hippocampus replays what that pathway labeled as the most important thing that happened all day. Here's something that rarely gets mentioned outside veterinary neuroscience. A 2024 study on targeted memory reactivation in dogs, researchers at a Finnish university cueing specific spatial memories in sleeping animals with subtle sensory prompts during non-REM sleep, found measurable improvements in next-day recall for the reactivated experiences. The sleeping brain wasn't passively recording the day, it was actively selecting, deciding what to retain at higher resolution, what to consolidate more deeply, what to discard. And across multiple studies measuring hippocampal replay in sleeping dogs, one category of experience was consistently overrepresented in what the brain chose to strengthen. Social bonding events, proximity to the owner, contact, play, the sound of a familiar voice. The twitching paw is tracking something. The eyes moving behind closed lids, are following a scene reconstructed with enough fidelity that the visual cortex cannot tell it from reality. When your dog's tail moves against the floor while they sleep, they are, in a neurologically specific sense, experiencing you. Not a warm approximation, not a vague familiarity. The actual hippocampal firing pattern of your presence, replayed by a brain that selected this as the highest priority information it collected all day. That's not anthropomorphism. That's what the data shows. If this changed how you see the twitching paw, the muffled sound, the tail moving against the floor while they sleep, subscribe. We look at what the neuroscience actually shows about animal experience, not what we tend to assume. What do you notice most when your dog dreams? The paw movements? The sounds? Something else entirely? Leave it below. Only someone who has sat quietly next to a sleeping dog can answer that question the way you can.